It's recording. And thank you all for joining today. And I also want to thank the Stem Cell Network and everyone here that supports us. Just a quick note, don't forget that the TMN 2023 is going to be hosted in Toronto, and please register. We're going to have our virtual human workshop at the end of it. So please join us. We hope you can join us in Toronto. So today I'm really happy that Naoki agreed to give a talk for our Bright series, and just to start a discussion, or at least to have you thinking about these evolutionary process and how we can use them in our understanding of the virtual human development, or at least to think about evolutionary forces and how they work in with response to development. For me, evolutionary forces work on a different time scale than developing system, at least the ones that we're looking in. But it is clear that these forces forged the mechanisms that we are trying to disentangle. So we need to sort of understand how or what changes were made across this evolutionary timescale that maybe they might give us some insight on what should we be looking in a particular system. And so, for instance, last time I talked about cell cycle and gene length from my work, and those are two different mechanisms that I tried to show are important in understanding cell diversity, and they might even be able to predict some of the cell diversity in a system. But what I didn't talk about is how these two systems actually co-evolved, or there is some data evidence that show that cell cycle and gene length co-evolved across many species. And what they see is that gene length actually increased in different systems. So if you look at the yeast, then C. elegans, mouse, and human, you can actually see certain genes had introns added to them. And this could be 1 million base pairs just added into the system. And just knowing that certain genes were targeted and increased across time, while cell cycle duration also increased, might give us insight into sort of maybe what happened. And maybe it also helps us understand what should we be targeting in a system. So I actually think understanding some of these evolutionary histories might guide us and to help us understand what changes were made and how were they important, and maybe we can use them to try to predict or even target certain cell types. So thank you, Naoki, and the floor is yours.
Thank you. Thank you for the great introduction. Actually, I have also been thinking about evolutionary forces has much less to do with the something happening during development, because natural selections are the main force, I would say, and then genetic drift, of course, but natural selections are the major force to shape the organism. And I was like, well, development may create or may generate variations and diversity, but it doesn't necessarily mean that we know how the organism would evolve. And then development may have much less to do to tell or to predict how the organism would be like. But our recent study suggests or implies that development may have pretty strong force to limit the organism to what they should be or what they become in the future evolutionary outcome. And I've long been studying this project in University of Tokyo, but I've recently moved to Research Center for Integrative Evolutionary Science. It's the sole institute focusing on evolution in Japan. It's been launched in last year. And I'd like to summarize my talk first. The one is repeated usage of same genes or genomic modules may have double-edged sword effect toward phenotypic evolution. So it has long been said, and then there are numerous numbers of studies that supported reusage of genes, like reusing genes in leg to beetle horn. They actually facilitated to evolve new traits like beetle horn. And this was the major effect known for gene co-option or gene reusage. But what we found is that reusage may also constrain diversification or limits the phenotypic diversification. And the second message is robust and stable phenotype may limit its evolutionary diversification. Like inertial force. So evolutionary theory, like Darwin's natural selections and modern synthesis, we usually consider what kind of variations are available for natural selections, like variety of phenotypes, and we select from them, and evolution occurs. And this was kind of a major strain or major explanations how the organisms would evolve. But one implication is that since the developmental system or the organismal system itself has limitation to create variety of hypothetical phenotypes, like branching tails or cats with wings, or huge head, or multiple legs. So since the development has much less limited potential to create phenotypic variation, they can only create limited numbers of variations in terms of hypothetical phenotypic variation with respect to hypothetical variations, and that may cause inertial force to the evolutionary outcome. And these two are the main message from my talk. And please don't hesitate to interrupt me when you have questions, even on chat. I'm happy to take it. And then I know that is supper time there, so please enjoy your supper if you have time. And then I hope you also enjoy my talk. So why did I jump into this kind of problem? Because I was interested in how we can predict the evolutionary outcome. Oh, thank you. Here comes feed. Let's see. Why do you think certain stages of development are much more selective than others? Implantation for humans. Yes, there are several hypotheses, and let me go through that. Yeah, that's exactly what I wanted to talk in this presentation. So basic anatomical features of animals appear to be difficult to change during evolution, but I was wondering why. Because, for example, it's just a hypothetical organism, but we feel this kind of organism may have less chance to evolve from human being in the future, even if we spend billions of years. And this is probably because we already know that the anatomical changes are rare during evolution. But this is only our assumption, and we wanted to, or I wanted to make it more quantitative. And supporting evidences are that body plan is somehow very conservative. For example, since the Cambrian explosion, like 500 million years ago, or 600 million years ago, every phylum lineage retains their basic anatomical feature. So no new body plan has emerged or even branched from the existing lineage. I was wondering why the body plan should be conserved in such a strict way. And this is the body plan for chordates. We all have head, dorsal nerve, notochord, laryngeal slits, and ventral digestive tube, and segmental structures. But why do we have to keep this? And I was wondering, there should be certain mechanism, but others also said that, well, it's just because it was adaptive. So I wanted to answer this. And indeed, by looking at more smaller evolutionary timescale, we see plenty of species that underwent coat color change or size change, or even when flattened, but still, it's pretty rare to have this kind of anatomical change. For example, turtle has shoulder blade in front of their rib cage, which is almost opposite to us because we have shoulder blade in backside of our vertebrae and rib cage so that we can move our arm like this. But turtles, they somehow change the position, and it's a very rare case. But even by looking at the developmental processes or during development, it's not topologically opposite. It's just how do I say, tilting, and connection itself has not changed. Like which bones are connected to which bone for which muscles or tendon has not changed. So it's just, it's not a complete topological change. So even turtle could not change drastically. So the reason is not clear yet, but based on the hourglass model proposed in 1994 by Duny de Beau, he said that this is probably because the body plan is conserved because of the mid-embryonic conservation. And since this body plan developing stage, we call it phylotypic period, but since this is very conservative, won't change during evolution or through generation, this would be the reason why body plan is so much conserved. But it's just how do I say it didn't answer anything, you know. I was wondering why body plan won't change, but the question simply shifted to why this mid-embryonic stage won't change. So it's just a shift of question, and then nothing has been answered. But still, this hourglass-like conservation has been supported not only in chordates, vertebrates, but also in nematodes, Drosophila, and other group of animals. But this was counterintuitive because when we look at the molecular developmental process, you know, it goes like domino toppling. When we yeah, sure, yeah, drop off, yeah, sure. We have recorded version. So since it's like domino toppling, the signaling cascades expands during during development. So it sounds like the earliest stage should be conserved the most. So early studies or early arguments without any experimental evidences, pioneering scientists argue that earliest stages should be conserved the most because it's responsible for following developmental processes. So the outcome should be earliest stages should be conserved the most. But this was not supported in the end, as the hourglass model has been supported. So during development, mid-embryonic stage was the most conserved stage. And let me briefly explain how it was supported. So what we did is to compare whole embryonic transcriptome. So there's going to be no morphological information. I sadly had to discard all those morphological information and compare the similarity of transcriptome. And my assumption is it reflects the similarity of transcriptome, whole embryonic transcriptome reflects a composition of homologous cells between different species or different developmental stages. And we calculated similarity and found out mid-embryonic conservation. So we also did detailed or minor improvements to our methodology, but let me skip this part. So first question was, so how long has this mid-embryonic period has been conserved? Is it like several generations or vertebrate-wide generation? So we already know that it's been conserved for each phylum, animal phylum, like chordates, arthropods, nematodes. But how can we go into smaller scale? And the second one is just like Nika made a question, what's the evolutionary mechanism of this conservation? And we collected 8 chordate stages from more than 10 developmental stages with, of course, biological replicates. And we could target or focus on turtle embryogenesis because we have plenty of local farmers in Japan. Yeah, I never tasted it, but it's a traditional food culture in Japan. And then thanks to the food culture, I could get access to turtle eggs. Then let me start from comparison between turtle and chicken embryos, which split at more than 250 million years ago. And since there's no equal developmental stages, I did in an old-to-old manner comparisons and found out these stages has highly similar whole embryonic transcriptome. So whenever the bar is there, please think that this has more conserved. These stages are more conserved than other developmental stages. So this is one of the results came out, even though they have pretty different adult anatomical feature and pretty different size in egg size, but still the meta-embryonic stage resembled each other, even morphologically. So what if I expand the evolutionary scale? So I added mouse to this, and the turtle chicken conserved stages were still kind of covered or overlapped with the amniote-wide conservation. And this was also true for when I added xenopus species, and even by adding zebrafish, it became a little bit blur, but still it was covered. The turtle chicken stages were covered. So which means that this stage has been conserved not only from split of turtle and chicken, but from way back to vertebrate evolution. But when we add the proto-chordates like Amphioxus, Cyona, the results were a bit weird. It did not follow the hourglass-like conservation. So I would say vertebrates is the origin of the hourglass conservation for this lineage. But this was weird because when we only focus on one-to-one orthologs, the meta-embryonic conservation was also observed for Amphioxus or chordate-wide species. So there should be something different between strictly conserved genes and whole genomic or whole transcriptomic conservation, including paralogs and genes that are presumably lost during evolution. But anyway, I was wondering, so I found that the conservation is not only for vertebrates or even chordates, but also to smaller evolutionary scale. And we also, our recent study supported that even within species, this meta-embryonic stage is conserved. And I was wondering why that should happen. Because vertebrates has experienced not only ocean, but freshwater, land, sky. Is it, does the environment has nothing to do with this conservation? I was wondering.
Can I interrupt you? Just
Yeah, sure, yeah.
ask a question, just to the previous figure where you had more, because some of the species had some conservation early on in these early stages. Is it because they shared, like, because I see some from this 2-cell stage, there's some hint where, zebrafish did not have any hint in the first 2-cell to 32-cell stage, but then that increased in their conservation. Is it because those genes were present at the time? Is that what we're comparing? We're comparing the number of genes that were present within the system at the same time?
Present, you mean
Like
expressed
expressed?
or?
Yeah.
Yeah, they're expressed, yes.
So the cool part is that you compared, let's say, 2-cell stage to a 10-cell stage. It didn't matter. You compared all the different connections, right?
Yes, exactly, yeah.
And as long as there was a hit of some sort of similarity, that was considered as a conserved hit. I guess I was really more, I just seeing those ones where they were like highly conserved in the early embryonic stages, like the first few cells. And I guess it was the Xenopus that had that? I just was curious to know, like, what is it that they had that caused them to be more conserved? Is it
The
just,
reason or mechanism, right?
or well, we're comparing expression to expression, right?
The genes, you mean?
Yeah, so did you have like, was there like a specific set of genes that, like, was it the Hox genes? Was it a particular sort of was it just the notochord? I just was curious to know, like, is there a specific sort of region that seemed to be causing this conservation a lot more, like giving it a
Or
higher hit, or did you see it sort of across tissue? Does that make sense? Sorry, I hope that
yeah, we actually looked for, is there any set of genes that contributes to conservation, especially for mid-embryonic stage? And we tried very hard to extract what kind of genes are there, especially for those contributing to the evolutionary conservation. But unfortunately, nothing special came out. And of course, developmental toolkit genes, like including Hox genes or morphogen genes, transcription factors, those are detected in the mid-embryonic stage. But these were also detected in early and later stages. So it was not specific to mid-embryonic stages. So I may have, we're recently trying to do this further with machine learning to extract what kind of gene set, or even by including a taxec data set, and extract what kind of network or potential regulatory elements contribute to the conservation of certain developmental stage or certain cell. Because I have no slides today, but we're also trying with single cell analysis and putting them into machine learning system to let them extract what's the content that contributes to conservation. But so far, much less have been detected. Did I answer to your question?
Yeah, no, I guess you didn't know. That's why I was curious to know whether there was a specific element that was creating this constant conservation, because that would be interesting.
Yeah, so honestly, we failed. Yeah.
No, I don't think that. I mean, you have a hit. It was just curious to know what is causing these the signal. Right. So
Yeah, thank you. And then it actually have another answer to that question. So for the first question, yes, from interspecies to subphylum level, or maybe phylum level. But the next question is, what's the evolutionary mechanism? And some said, well, development is like intergenerational scale, and evolution is intergenerational or cross-generational scale. And does it really have something to do with this conservation, especially those for millions of years? Some said no, and some said yes. And I was a bit worried about stepping into evolutionary mechanism, because it's a history of hundreds of millions of years, and then we only have species that are available now. I was like, should I really jump into this? But this can be the main question. So yeah, let's do it. And that was the reason we jumped in. And supporting evidences is that vertebrates, especially vertebrates, have to accomplish development safe and sound every generation. So it's a thought experiment, but what happens when you get rid of development from animal? So without development, like ESL can be passaged and then continue to passage for generations and generations without development or without making an adult form. But it can easily acquire drastic mutations within like dozens of generations. But this will not happen during our generational change. So it kind of implied, at least to me, that when you get rid of development, the genetic system may have much less constraints and then goes more freely to change their chromosome numbers and accumulate more mutations. And we also know that even germline cells have to survive cell competition. So there are very strong cell competition during the development. And we also have, when we die during development, or when certain organism or individual die during development, the individual has no chance of creating next or further generations. So it's kind of filtering process. And the first the very major idea to explain this conservation, it's one of the most famous and classic one, was that negative selection is working toward here, that's in embryonic stage. Because when you add mutations or even noise, environmental noise, to this developmental stage, it's just an assumption, but they predicted that since this stage is very fragile, it's very easy to die or become lethal phenotype. And then this leads to evolutionary conservation. But what we found by doing, for example, by adding UV irradiation at the early stage and see the survival curve, and we found that not the phenotypic or this medium embryonic stage has the biggest drop in survival curve, but it was almost flat. But the drop was found a little bit earlier, like gastrulation stage. And this was also true for, this is for zebrafish and for xenopus. So the phenotypic period was not that fragile. And UV irradiation may lead to lethal phenotype by accumulating wrong developmental processes due to mutations acquired in the early stage. So we also added perturbations stage by stage. But earlier stage was more fragile due to our results, for not only for zebrafish, but also for xenopus. And the hypothesis that medium embryonic stage is the most fragile stage has not been supported by our results. Sorry. And the next idea was, well, it's probably because there's no or less positive selection acting toward mid-embryonic stage, because earlier stage or later stage has to adapt to environments, variety of environments. But mid-embryonic stage is just a stage that goes through and has no positive selection acts toward it. But it's been done by Zhao et al. In different group. They, using C. elegans, they nicely demonstrated, well, they could artificially create environment without positive selection. And they have done experimental evolution. And they found that even without positive selection, the mid-embryonic stage remains conserved. So this hypothesis was not sufficient to explain. Then what would be the potential mechanism? Then we came to pleiotropic constraint. This first began by one message from my collaborator. He said, how about extracting gene sets that are specifically expressed or downregulated during meta-embryonic stage? And I was like, I don't know. Can we really explain something about hundreds of millions of evolutionary years from gene sets? I don't know, but let's try. And what happened was that my colleague or collaborator told me, well, my hypothesis was wrong, because he showed me that genes that are specifically upregulated or downregulated was much less than later or earlier stages. So unexpectedly, less stage-specific genes were found at meta-embryonic stage. And we talked over those results, and then, well, it's probably because meta-embryonic stage has abundant pleiotropic genes, genes that are expressed everywhere or expressed in a variety of developmental stages and tissues. And actually, we looked for features of this pleiotropic genes that are expressed in a variety of tissues and developmental stages. When they have mutations, they tend to die. And when if they are expressed in more stages, mutations tends to be more lethal. In addition to that, I analyzed the database, and they have been suggested to have more protein interactions. So when genes were expressed in more stages, it's a log scale here, but expressed in more developmental stages, they tend to have more counterparts to interact. And what is important, when certain developmental stage is enriched with pleiotropic genes, they tended to be evolutionarily conserved.
Can I interrupt again?
Yeah, sure.
I just want to know, like, what is the difference between a pleiotropic genes and a paralog? Is that like what is it? Yeah, I just mean, maybe you can define what a pleiotropic gene is for me.
So pleiotropic genes here we define as genes that are expressed in stages more than 50% of the whole sampled stages. So let's say we detected or analyzed 10 developmental stages, and certain gene was expressed in more than 5 stages. The genes were defined as pleiotropic genes.
Okay. So, okay. So I forgot. So it's more about like the idea of a single gene can apply to more than one like phenotype or a trait within the system.
Exactly,
yes. And then for you, it was a gene that you can see across multiple stages.
Yes.
Okay. And
also, yeah.
Yeah, sorry. So then in this case, you saw these genes not neither downregulated nor overexpressed when it was in the middle stages. So they were sort of like waiting in between, like,
or detected everywhere.
Or averaged. Yeah, I wasn't sure what it. Right. So just to push kind of the thoughts, this is a more of an average analysis, right? Is that yeah, what would you think would happen if all of a sudden now you're looking at a single cell level? Do you think that you might be able to distinguish certain like lineages with high access to these genes and other lineages low, and you're actually seeing, instead of seeing this like diminishing result, but actually seeing a segregation of lineages? I mean, I'm just imagining, like, I know like we see one picture here, but what if single cell data was able to not reveal that it's more of a everybody looks the same, but maybe this is a true segregation of lineages, and all of a sudden everybody's down a certain track. When you average them out, we're down in the middle, but really when we think about them, they're more diverse than we than they need to be. And maybe that was what makes them robust. I don't know. I just wanted to know, like, I mean, how would you imagine them, if you change the resolution of your.
Yes, and then that's exactly what we are expecting to have from our single cell, single nucleus RNA-seq results. And we're expecting that, for example, later stages may have, even though they have like average expression level, but I wouldn't expect all the cells expressing the pleiotropic genes, but having like certain specific tissues or cell clusters are expressing, but some are not. And those could be the reason for this results. But yes, that's the results we're expecting when we change the resolution to single cell level.
Okay, interesting.
Yeah, but we still have to know then why that have to be have to cause evolutionary conservation.
Actually, I have another, sorry, just more about to get the question.
Yeah.
I know you're saying that they're conserved. What if they were selected? Like, I know you see like death in early on, but what if these genes are selectively selected in these early stages and it's really not a conserved system, but they won and now we're just watching what wins? I'm just thinking, if you have like 100 embryos and now we're really only watching the ones that successfully made it to that point. And so what distinguishes between a selected mechanism through development versus an evolutionary selected mechanism?
So what do you mean by selected in the former?
Right. So we don't think about a development as a constraint, but what if we have like checkpoints within development, and these particular like when you get to a certain time point, there's a checkpoint, and maybe all the ones that didn't make it were weeded out. So it looks like there was this evolutionary constraint, but really they just didn't make the checkpoint. So maybe there was like this boost of desertification. Think about the Cambrian explosion, right? There is the idea that there's a million body plans and only certain one of them made it through. And so what if this early development, the crazy chaos is early on, and then only the ones that make it past the checkpoint make it through? And so how do you distinguish between making it through checkpoint versus a constraint from evolution?
That's difficult to answer, of course, but the extant species may have succeeded by creating this kind of checkpoint or constraint. It's just my assumption, but they succeeded in combining certain organ modules, for example, to create variety of phenotype. But for those that did not create those organ module interactions or body plan developing phase, they still remains to be more, how do I say, sort of, it's a misleading term, but primitive structure or primitive organismal structure. So I would say the checkpoint itself made them to succeed, but due to that, they also have constraint because of the checkpoint. That's my kind of assumption at this moment.
It was just more of a just push the thoughts. I was just curious to know. One last question before I get to, why didn't you pick sort of like a non-chordate species, let's say the sea urchin, or I mean, they're like, I think they're a good developmental model, but they also have species that are, close to the Precambrian. I know there's fossils, but I just meant like, would you think that you'd see some of these similarities?
Yeah, we actually did. And then we actually sequenced 3 echinoderm species and then did a whole embryonic transcriptome. And they also showed hourglass-like conservation.
Even like, so just, is it before they metamorphose or like in the juvenile stages or after they metamorphose? Did you see it?
Around right after gastrulation stage, but not the stage where pentameric symmetry establishes, because that is said to be the body plan for echinoderms.
Right.
And our assumption was that that stage, pentameric establishment, that would be the stage which shows the most conserved signals, but the stage wasn't. So I would say
Oh, interesting.
body plan for echinoderms may not be pentameric, but more early or features that can be observed at earlier stages.
Right. I mean, if you look at the heart urchin, they are like, they're not pentameric, and only certain species are pentameric. And
Mhm.
there is the idea, like, within each, let's say, column, or
Mhm.
you want to look at the pentameric, it's a single Hox gene that seems to form the system. So you can look at it as a single. And if you look at we don't need to get into echinoderm, although doesn't love echinoderms. But I was just curious to know, like, because they have species that go to precamp, like up to the explosion, and you might be
Yes,
able
yes,
to get some body plan.
yes, exactly. Yeah, thank you. Yeah, we actually did that. Yeah.
Okay, great.
So it's just a hypothetical excellent I say hypothesis, but pleiotropic constraints has been proposed in 1999, and then the reason why pleiotropic genes may lead to constraints, we still do not know the exact mechanism, but one possibility proposed by Fritz and Gallus is that because she was interested in why mammals have almost always have 7 cervical vertebrae, even though some species have long neck, but some have short neck. And she proposed that it's probably because important genes like Hox are connecting variety of biological systems or processes. And when you have mutations to Hox genes, it means that everything will be disrupted. So this makes the variety of developmental processes or biological systems being connected or chained to each other. And this chain makes them difficult to change independently during evolution. And this was the idea. And as a support for this, she performed meta-analysis and found out human patient with extra rib, for example, has very higher risk of raising cancer, 50 to 200 times higher than people who has no extra rib. So that's potential mechanism for pleiotropic constraints, like why stages or processes that have pleiotropic genes ends up with evolutionary conservation. But when we submitted this manuscript, the reviewer said, well, are you trying to overturn everything we have been accumulated? For example, as I just briefly explained in the introductory part, when you reuse the genes in different tissues, for example, genes expressed in legs were reutilized to facilitate the evolution of novel traits, new traits like beetle horn. And there's not only for beetle horns, but there are accumulating numbers of reports supports that gene reusage is more easy because it's much easier than creating new coding gene. It's just to change regulatory region, and there's going to be a few mutations to make this happen. Let's say there's upstream transcription factor, and when you change that little bit of regulatory sequences, then you can reuse all of them, all of downstream genes to a new region. So our understanding at this moment is that since the regulatory changes are easy to make drastic changes, it may sometimes lead to facilitation of evolving new traits. But our results is contradictory because when we reutilize the genes too much, then it may lead to pleiotropic constraints or evolutionary conservation. And I'm not trying to deny this, but I would like to say they have, or reusage of genes have double-edged sword effect. But exact mechanism for the pleiotropic constraints remains to be clarified, because what we found is not the conservation of gene or genomic sequence itself, but we observe conservation in expression level. So we have to, there are like some more questions we have to solve to really say that pleiotropic or reusage of genes leads to evolutionary conservation. So I was pretty much satisfied, but still there was another idea to explain the conservation, which came from physics background. An idea was that when certain developmental stage were stable, stable means here by keeping the same phenotype without mutations or without any environmental fluctuations or perturbations. So it's just like stable phenotype without any noise, or from random noise, stable against random internal noise. And theoretical studies predicted that this stability correlates with evolutionary conservation. And I wanted to test this. So would this stability correlates with evolutionary conservation? Sorry, let me skip this one. So theoretically, we have to deal with the clone individuals, embryos, to test this, but it was hard to do it in vertebrate systems. So we used inbred medacoline, brothers and sisters, like gender matched siblings. And from same parent, same water condition. And we also tested, in order to measure the stability or phenotypic variation, we removed technical errors and normalized and measured phenotypic variation or fluctuation observed for each developmental stage. So this was our expectation. We may have more stable phenotype or stable feature for mid-embryonic stages. And here's the actual result. Just like we expected, mid-embryonic stages showed smaller fluctuations. They were more stable. And this, of course, correlated with evolutionary conservation. When we, there was a closely related lines in Medaka fish, and we compared intraspecies comparison and found out mid-embryonic stages here are conserved, and this nicely correlated with the stability. So not only for developmental stage or bulk RNA-seq transcriptome, we also looked for stability of gene expression, like gene by gene stability. And by applying normalization to get rid of technical noises, and also expression level matters, because whenever the gene expression level is high, it tends to show more stable expression level. So we also normalized that, but still we found kind of positive correlation between interspecies diversity and stability, gene expression fluctuation. So the fourth idea may also be supported. So we were, okay, stability. And the first one here implies that it's the mid-embryonic stage is robust because it was more strong against mutational and environmental changes. So the mid-embryonic stages may be more stable and robust than earlier and later stages. But I was wondering, does it have to do with pleiotropic constraint? And we collected variety of tissues and developmental stages and analyze correlation between pleiotropic expression, and stability. And I would say very weak signal, very weak signal was detected, but they won't contradict to each other, at least. That's my interpretation. Very weak correlation here. So I would say maybe number 3, pleiotropic constraint and robustness, stability would not contradict to each other. And finally, this is going to be the almost the final slide, but what if we cross with distantly related lineage and see the outcome with the stable genes or genes with stable expression in the ancestral generation? We crossed for 3 generations and checked if stable stages or stable genes tend to be more stable after 3 generations of crossings with distantly related lineage. And we found that, yes, stable genes, or no, stable developmental stages tended to be more conserved after 3 generations, and stable gene expressions remain to be less variational after 3 generations. So it's possible that stable genes or stability could be the cause of evolutionary conservation. And as a whole, how do these imply for the mechanism of the evolutionary body plan evolution? Sorry. So as I briefly explained in the introductory part, stability or robustness directs the evolutionary outcome, because when you have limited variations as a target of natural selection can only pick up from variation from here. They can never pick up something that are hypothetical or that never existed. So the developmental bias is one of the strongest bias toward evolutionary outcome. And that bias emerges from stability or robustness of the system. So for example, we have to test more, but another possibility would be redundancy of the genetic system. So whenever you have multiple redundant system, even though you corrupted with one system, you have another backup, which means that development can proceed normally. So those robustness and stability, I'm not really sure for how stability are implemented in organisms, but these stability and robustness limits or has less potential to create phenotypic variation. And that leads to evolutionary inertia. The less variable players appear, less diversity, and is the main message of my whole talk today. Oh, finally, we also analyzed the potential regulatory sequences or through ATAC-seq and found out after the mid-embryonic conservation or mid-embryonic stage, evolutionarily new regulatory sequences are more activated. But we're still not sure how we can reconcile with it doesn't contradict with the hourglass model, but how we can explain both regulatory aspect or regulatory results we obtain and transcriptomic results we obtain. So we still do not know the exact correlation, but tendencies that later stages uses more newer regulatory elements. Although we didn't check if it is really a regulatory elements or not. Insulator, promoter, I don't know. It's just an ATAC-seq results. So remaining question is, we still do not know the exact mechanism of plagiaristic constraints. And there still remains a gap between conservation of whole embryonic transcriptome and body plan development and developing phase. I would say, in other words, transcriptome and anatomy or structure. There's a huge gap because we only did bulk RNA-seq. Although we observed similar morphological feature at the meta-embryonic stage, but we don't know the logic behind these conservation. And we still cannot make quantitative predictions toward phenotypic evolution because we still do not know how strong is the inertial force is toward evolutionary diversification. So there are plenty of things to do, but in analyzing, well, after succeeding, how stability and robustness would end up with evolutionary conservation, we may be able to come up with new theory to make a prediction, not only for prediction, but why we look like this. Is it really just because we were adaptive, or were there strong limitations that we cannot make, let's say like wheel-like arm or something like that? So limitation and adaptation, and we want to know the balance between these two. So this is my talk. And I would like to thank my all collaborators and happy to take questions. Thank you.
Thank you so much, Naoki. That was great. Are there any questions? Because I have a few.
So for Nika's question, why do you think certain stages of developmental are much more selective than others? Our first assumption was that meta-embryonic stage is more selective or being selected by negative selection, but it was not. Gastrulation stage was more selected, and it's also known that implantation or even the gastrulation stage that comes after that is one of the limiting step for development. And we're not sure why those became the more selective stages than other stages. But these may have much less to do with the evolutionary conservation somehow.
You don't think there could be a consequence? Like once you select for gastrulation or implantation, you are, limiting or constraining the potential outcomes, right? So how can you separate which caused what? This is kind of my question as before, like once you have this selection at a certain checkpoint, which in this case would be gastrulation or implantation, how do you know that this constraint is not just a result from these selective processes? You're causing your own drift, essentially, by ensuring that the ones that pass their checkpoint are gonna get through. So how do you know it's not necessary, it's just a selective process versus a conserved process? This is kind of why I said
yes, my assumption is those selective pressures has less power or less force to make the developmental stage conserved when compared with the stages that have less potential to create phenotypic variation. Did I answer to your question?
Yeah, I'm still like iffy about, what's causing like the chicken or the egg problem, right? So for you, you're assuming that there was a selective sweep and this kind of stayed across all species, where I'm thinking maybe it wasn't necessary to selectively sweep because there's already inherent sweeping that keeps occurring over, and that's kind of what you're observing right now. Like you're observing a current selective process versus a conserved selective process.
Exactly. Yes. It's also true, possible that this is just a consequence, and then philanthropic constraints or stable or robust stages appear just because of natural selection. That's possible. And we're still not sure how we can distinguish whether this was the cause or this is just an outcome of evolutionary selection or selection against or through generations.
Yeah, I mean, I think there is evolutionary mechanisms that guided, like I said, but I'm just wondering, it's hard to distinguish between a current process. But okay, so back to this, like, because I just want to kind of focus on your work. So in the hourglass model, I think it's quite interesting, but what I'm still not convinced is how do we know that it's already there's regulatory mechanisms that try to ensure development is happening at some sort of, I mean, at least I feel if there is pressure anywhere, it's pressure during a developing embryo. That's when you want to do the least mistakes. And I'm not saying that there has to be negative selection. I'm sure that kind of swept through, but once you're putting some resources in and all of a sudden the cells are making these decisions and trying to propagate in order to make this viable organism, I think there's so many mechanisms that are working hard to ensure that it's not just a stochastic process. I'm not saying that it's not stochastic. I'm assuming, let's say everything is stochastic, but now all of a sudden development is working hard to ensure robustness and ensure all of these things. So as far as I'm concerned, the question is, what are causing these robustness? But here you're kind of saying that evolution already did the robustness part for me. And it's not the mechanisms, let's say the GRN and all the other things in between that are working hard to ensure that this is not a stochastic event.
So the question will be the robustness and stability has been made through the selection, or
well, yeah, like I just meant, like, so when you, at least from my evolutionary work or the work that you assume that there's stochastic effect in evolution and natural selection is selecting from the current state, and if you were to rerun this whole system, it's not going to look the same. Even Gould said it, like if you were to go back to the Precambrian explosion, we're not necessarily going to have the same body plans that we see right now. So that was just sort of like the stochastic selective force. It could have something else could have won. But he also, and I believe in one of his books, he does argue that there are particular constraints to body types. Like regardless of what you think could be possible, there are constraints, and you can only go down certain paths irrespective of what you think could happen. But now I'm thinking, okay, we have these evolutionary forces that sort of made these species what they are. But now we have development, which is something that, individually, each individual is ensuring that they're going to propagate. And so now there's like this individual selective event. And now our cells, or my cells, are going to try to create as much of a viable organism as I can. And so in itself, there's these robust mechanisms that have to be turned on, irrespective of what my, evolutionary stealth state is.
So
right?
it's
Because
like
cancer tells us stochasticity still happens and mutations still happen and mistakes still happen. So obviously development is trying to override all this.
So it sounds like the robustness or stability were almost inevitable for animals to
survive.
Right,
like, or to propagate. Like I'm guessing as soon as we became multicellular systems, we had to control these wild cells. At least my assumption is cells are individuals who want to take over, and if they were bacterial cells, each cell is to itself. And now all of a sudden we're creating these multicellular groups. Sometimes we cooperate, sometimes we defect, and so development is this cooperation in hopes that we're all going to work together. So the evolutionary forces came to ensure cooperation, and there's so many different mechanisms that show cooperation can evolve. I just meant like it's hard to, like for me, I'm thinking cooperative forces are kind of working versus then conservative constrained for just a particular time point, which seems to be an interest, like, because you're telling me only a particular time point and all the species is conserved, but everything else is chaotic. And it seems really hard that evolution pinpointed a particular time, but allowed the cells to stay chaotic everywhere else.
So I totally agree that it was almost inevitable for animals or multicellular organisms to have stabilized stages or stabilized or features or robust features. And it happens multiple times probably because cross-phylum comparison does not support the hourglass model, but they independently have been supported in different lineages like vertebrates, echinoderms, nematodes. But when we try to compare them across phyla, then the mid-embryonic stage conservation disappear. So it seems like the stability and robustness were inevitable for them. But what I'm thinking is, I'm not trying to say the earlier and later stages are more stochastic and can change anywhere they want to go, but they somehow, for some reasons, which I do not know, but for some reasons they were mid-embryonic stages were more stabilized, and then became more robust. And assumption, I do not have clear answer or evidence to this, but my assumption is the set of organ modules or system module observed at mid-embryonic stage or body plan developing stage was so much successful. And then that's, I think, the reason, or so much important. And then that's the reason I think they have been more stabilized and robust and conserved. So it's almost like, yeah, vice versa. And then causality, honestly speaking, causality is not clearly defined in mathematics. Even mathematics just gave up to clearly define it. So it's hard to clarify which was the cause and which was the outcome. But still, I agree that stability, robustness was inevitable. But why that stage? Not really sure yet.
Well, I think it's cool. I mean, there must be a way that we can generate an evolutionary model that kind of tests the potential, like, allowing all stages to be robust, all stages to be chaotic, and see which ones are more likely to evolve like a stable state, and under which conditions would you get this hourglass again. Like I mean, that's a different, but that's just like a thought.
Yeah, but actually, one postdoc in RICAN actually performed that kind of theoretical study simulation and found out that mid-embryonic stage somehow becomes conserved, starting from the stochastic line of cells, hypothetical cells. And so, yeah, we're trying to clarify not only from something we observed, but from theoretical approach. And I'm very curious, what would be the reason for this conservation or stability?
I mean, it's necessary, and it's obvious that it's another level where development is ensuring you're going through the right steps. But it's quite interesting, and I love that you've explored it across all these different phylas and classes and species, and I hope everybody appreciates that these kind of studies are gold mines for our knowledge. So, and it's a lot of work. It's not an easy amount of work. So I definitely think it's wonderful. Thank you.
Thank you.
Are there any other questions? I don't mean to bug Naoki longer than necessary, but I do find it fascinating. No?
I'm just starting my work today, so it's nothing for me, but you guys are.
Well, I guess, I mean, before like we wrap up and stuff, I just hope everybody understands, although we are trying to model the human development, it is these key elements that we see that are maintained across species that will be important for us to put in our model, because those we will know the conserved states will be points that right away we know are we won't have to question once we identify them. So once we know what is conserved, we might be able to know what mechanism led to that, and maybe it gives us insight in what's happening to these cells. And hopefully then we have a model that can extrapolate outside of our knowledge state. But these type of studies are key to understanding what happened across time and maybe what made humans human, right? So what were the changes? What makes it, I mean, I don't know if you did that, but let's say a mammal, I don't want to go, just in case you don't have a human species, but let's say we picked a mammal, which is the closest to our human, mouse, or some sort of a monkey, and then we identified what made it different from all the other species. And so what makes the monkey across these stages, just kind of blindly understanding where is the constraint that, or at what point does the monkey become different from all the other systems? And maybe those will be also key target points that will help us, access humans, because we cannot access human cells after certain time points. We cannot predict, but understanding what is the same across species will help us, add to the unknown, and understanding what makes it different will also make it more human in our system. So I hope everybody sees the benefit. And maybe, I don't, but maybe you can let us know what you would like to see in a model. How would we push this system, and what would be a virtual human model in the future? So
I would love to see how stability and robustness would be implemented or can be detected from the model. And talk for planitropic constraints and those robustness we found. I think we were lucky because we focus on vertebrates, but what if we focus on planarians, for example? They can simply cut off their, part of their body and expand and then create next generations. And then they don't have to go through developmental processes. So, which means that they can skip, even though some major parts of the body went lethal, but some part can create next generation, which means that developmental process is not always necessary. So I was lucky to find out these results because I focus on only those who starts from fertilized egg to adult to create next generation. So yeah, I'm thinking of how those asexual reproduction and sexual reproduction leads to different outcome in their shape. For example, genomic sequences of planarians were so difficult because they had enormous amount of mutations, even within one species. And I guess it's because of the developmental system itself. So I'm curious about how these asexual and sexual reproduction, especially development, when species have to always go through development, what kind of limitations would result in?
did you try to sea cucumber? Because you have an echinoderm, section.
Yes, we also had sea cucumbers. Yes.
I mean, I don't know enough about the sea cucumber. Doesn't it have a capacity to sort of, disintegrate and come back together? Did you find anything different? I'm not an expert in sea.
Not really. Unfortunately, I do not have the echinoderms here, but not really. They simply showed the hourglass-like conservation, but the stage was not the pentameric developing stage.
Okay, because I remember my PhD supervisor telling me about sea cucumbers having the capacity to sort of, if they're stressed, they disintegrate, or at least separate their spicules and then are able to come back together at some points. So those would be and interesting.
But planarians, they're much.
Yeah, no, I meant like you already had echinoderm, phyla, and maybe just adding one more echinoderm to the comparison. But it's interesting they didn't have that.
Oh, by the way, I don't want to talk for long, but I also did a microchimerism study for a long time, and human or placental mammals are known to have maternal cells inside their body. And we recently succeeded to get rid of these maternal cells from fetus. The fetus immune cells showed more activated status. So it's possible that maternal cells are suppressing fetal immune system. And in other words, from more general perspective, development would not start only from single-celled fertilized egg, but some cells from mother also participates in her development, especially for placental mammals and human. And the cells has been detected in variety of tissues and last for the rest of our life. So it's a cell level epigenetics, and it's also happening in our body. Not only the epigenetic markers like methylation or something like that, but cell itself is also inherited to our body. And development, our body is somehow coping with those non-self cells to create our body. So I'm not sure how we can, incorporate this idea to the model, but it's just one research topic from our recent study.
How do you link that to the maternal zygotic transition? I know humans don't have a long maternal zygotic transitions, but a lot of other species do that. Do you think that there is a link to, do you know what I mean? Like, so there's the idea that there are maternally injected transcripts already present in the zygote. And I mean, I don't know exactly about all the species, but I was under impression that humans had a short transition, like, so there's very little maternal to zygote, but zebrafish, fruit flies, and xenopus tend to have a longer version. I wonder, does this like compensate for, or do you think that's just a separate mechanism to ensure that not self is accepted?
I guess that's the reason for those. That's the major role of these cells, maternal cells, to avoid fighting each other immunologically during pregnancy, because the cells migrate only after E12.5 or E13, which is way much later than the zygotic maternal genes, maternal effect genes.
So they are sort of like almost eaten by the fetus, or like ingested, or sort of like, where are they, like, how do they get used? Like, I'm just,
yeah, it's not known, but there are two pathways, and one is through placenta.
Okay. Yeah.
And surprisingly, one is from breast milk.
Okay.
So we somehow acquire maternal cells from breast milk, and they actually reside in our body and get stabilized. And then, I don't know, in some niches, they remain, probably they propagate and remain for the rest of our life.
Interesting. Well, thank you. This was quite enlightening. I thought it was wonderful.
So you have mother in your body too.
Oh, I had no doubt. I mean, I'm guessing, I mean, you just made me think a lot. I'm just guessing that probably, like, versus breastfeeding, not breastfeeding. And then there's people who have later onset of certain allergies. I wonder if it's like the depletion of these cells versus not, like, it could be an interesting loop to go down thinking about it. But that's definitely, I guess once we are able to generate a virtual human, we have to think about the other environmental factors that affect how it responds to But I guess our first goal is to get that first.
Yeah, sure, yeah, I agree, yeah. Thank you, yeah.
Well, thank you, and I hope everybody else enjoyed it as much as I did. And if you don't mind, I would like to have some of your papers. I found one of them, but if you don't mind, like sharing, and then we can read your published papers, please, Naoki.
Yeah, sure. Which one? The evolutionary thing or microchimerism?
All of them,
All
if
of
you
them?
don't
Yeah,
mind. The
yeah,
ones
sure.
that are published.